Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue our state test review. We're going over the entire biology curriculum, hitting all the high points so that when you get to that state test at the end of the year, you'll know what's going on. So let's go ahead and get the ball rolling and see what we're talking about today. Okay, so here we're mostly talking about meiosis, and we're going to compare it to mitosis. Okay, and we're going to talk about why this is so important when it comes to reproducing in organisms. Okay, so that's what we're talking about today. We'll go ahead and get the ball rolling. The first thing I want to do is talk about these important vocabulary terms, because this is something that students tend to have a problem with, and if we go ahead and clear it up now, that's going to make the rest of the lesson a little bit easier. So what I want to talk about is the difference in between body cells and sex cells. Okay, so most of the cells in your body are known as body cells. Another name for that is somatic cells. Okay, these somatic cells, we call them diploid. What does it mean to be diploid? If you're diploid, these are cells that contain two sets of chromosomes, one from each parent, and we often write that as 2N. And so the example here is when we talk about how many chromosomes are in our cells, we often say 23 pairs. And that's what I mean when it says 2N. We often talk about how many pairs of chromosomes there are. So N would represent the number, and then 2 is telling you to multiply because they are coming in pairs, right? Okay, so let's compare these body cells to sex cells. So every cell in your body is a body cell except for your sex cells. And there's only two types of sex cells. In men you have sperm, and in women you have eggs. And these are known as gametes. So anything that's not a sperm or an egg cell is a body cell. And again, another name for that is a somatic cell. Okay, so sex cells, again, another name for these are gametes. These are sperm in men and egg in women. Okay, these cells are said to be haploid, which means that the cells that contain these only have one set of chromosomes, meaning one chromosome from each pair. And again, we're representing this as just an N. So your sperm cells and the egg cells, those have 23 chromosomes. Not 23 pair, just 23. And that's what we mean when we represent it as N. It's the number of chromosomes, but we don't write it in pairs. And we're going to talk about why that's so important and how that happens as we go through the lesson today. Okay, so the next thing I want to do is we're going to compare and contrast meiosis versus mitosis using this table. So I want you to kind of think about these as we review them. Okay, if you can do this, that's a good sign that you kind of know what's going on. And this is still important to review, but you remember the important stuff out of here. Okay, so let's compare mitosis and meiosis using this table. Okay, so the first thing that we're looking at here is at the very top of that list, it says number of daughter cells. Okay, well, what are daughter cells, first of all? Okay, well, remember that the cell that you start with is called the mother cell, and the cells that you end with are called the daughter cells. So in mitosis, how many daughter cells do we produce? We produce 2 daughter cells, right? 1 turns into 2. Okay, in meiosis, how many daughter cells do we produce? We produce 4. That's right, 1 cell is going to turn into 4 cells in meiosis. Okay, how many numbers of cell divisions do we have in mitosis? In mitosis, the cell only divides 1 time, right? Because you're starting with 1 and you're ending with 2, so you're dividing it once. Okay, how about with meiosis? How many numbers of cell divisions are there? In meiosis, there are 2 cell divisions, and again, that's because you're starting with 1 and you're ending with 4, so you divide it twice, right? Okay, how many chromosomes are in the daughter cells? In mitosis, how many chromosomes do you have? You're going to have the same as the parent. So in humans, if the parent cell will have 23 pairs of chromosomes, and mitosis, the daughter cells will have 23 pairs of chromosomes. How about in meiosis? Okay, in meiosis it's a little bit different. You have 1/2 of the parent cells. So in meiosis, if you're starting with 23 pair, you're going to end with just 23, right? Because you're going to have half, okay? So let's compare the daughter cells to the parent cells. Are they going to be similar, identical, or different? Okay, in mitosis, if you compare the daughter cells to the mother cell, they are going to be identical. Again, remember, mitosis is used for growing and repairing cells. So imagine you get a wound on your hand and you scraped a bunch of the cells off the top of your hand off. Well, mitosis is going to be the ones that are responsible for replacing those cells. Don't you want them exactly like the cells that started, right? You don't want them like, you know, a different color or looking different or a different type of tissue there. You want them pretty much the same. So we want the cells that we're ending with to be the same as the cells that we're starting with. Let's compare that to what happens in meiosis. Okay, so comparing the daughter cells to the parent cells in meiosis, the cells are going to be different. And this is a good thing, right? I love my parents, but I don't know if I want to be exactly like them, right? Think about you and your siblings. You and your siblings all have the same parent, but you probably don't all look just alike. Wouldn't that be kind of annoying if you and your older sister and little brother all looked exactly alike? That'd be awful, right? And so what's happening here is we want these cells to be different so that when we get a sperm and an egg cell, they're all different from one another. That's going to increase the variation in our population, and we're kind of shuffling the deck up. So you get some of your dad's genes and some of your mom's genes, but your brothers and sisters got different combinations of those genes, right? Okay, last thing on this table, let's look at that bottom row. This has the type of reproduction. So think about the type of reproduction. For mitosis, this is going to be asexual, right? Because we're starting with one cell and we're going to end with two cells, so there was no sex going on here. With meiosis, the ultimate goal here is that this is how we can reproduce sexually, right? We're making our sex cells, we're making our gametes, and that's going to help us make the next generation through that fertilization process, okay? So again, we're comparing and contrasting mitosis and meiosis here. If that kind of makes sense, that's a good sign that you remember this stuff pretty well. Okay, so what's going on here, okay? So through meiosis, we want to take one of these cells, a cell with 46 chromosomes, a cell that is somatic, a body cell, a cell that is diploid, and through the process of meiosis, we're going to turn that cell with 46 chromosomes into a cell with 23 chromosomes. And that cell with 23 chromosomes, we could call that a gamete, we could call it an egg or a sperm cell, we could call it a sex cell, and all of these cells are haploid. What I really want you to notice is that the haploid cell has half as many chromosomes as the somatic cell, right? These are kind of all words for the same thing, and these are kind of all words for the same thing. So why are we going through this process? What's the deal with why are we trying to do this, right? Well, think about it, okay? The whole process here, the whole goal is fertilization, okay? Why do we go through the effort of dividing the number of chromosomes in half? The reason is so that when an egg and a sperm come together through that fertilization, the normal number of chromosomes is restored. So now we've got an egg cell with 23 chromosomes, a sperm cell with 23 chromosomes. When they come together, we're going to add them together, and now we have a zygote with 46. And that's what we want. We want the cell with 46 chromosomes. If we hadn't divided them in half, then this number would be a lot higher, right? So that's why we have to do meiosis, is to divide the number of chromosomes in half. Okay? Great, so what I wanted to do next, this is just a pictorial overview of meiosis. I'm just going to go through it really quickly here. We just talked about mitosis, and the good news here is that meiosis and mitosis, they sound a lot alike, and the process is pretty similar as well. There's just a few key differences that you have to remember. So one thing, what's different about meiosis than with mitosis? First of all, you might realize that there are 8 phases instead of just 4. And that's because we do PMAT twice. So this is prophase 1, metaphase 1, anaphase 1, telophase 1, and then we do it again. This is prophase 2, metaphase 2, anaphase 2, telophase 2. If you remember everything that was happening in mitosis, the same thing's going on here. In both of these processes, the nuclear envelope is going to disappear, the chromatin is going to condense into chromosomes, and the spindle fibers start to form, just like in prophase of mitosis. In meiosis 1 and 2, just like in mitosis, the chromosomes start to line up on the middle. If you can just remember middle and metaphase, you'll have what you need there, right? Anaphase, same thing. The key word here is apart, right? Away from each other. The chromosomes are being pulled away from each other. And then in telophase, the word I like to remember for telophase is 2, because you can actually start to see that there are 2 new cells. So up here you can see how this is kind of forming into 2, and down here you have 4 total, but one of them is dividing into 2 and the other is dividing into 2. Okay, so what's really the key difference here? There's just really 2 phrases that you have to remember, and if you remember these, you'll almost always be able to tell the difference in between meiosis 1 and meiosis 2. Okay, and it has to do with this combination of chromosomes up here. So take a look at what's going on here, okay? And that top left for prophase 1. You see how it's 2 chromosomes right next to each other, 2 X's right next to each other. Okay, we call that a tetrad or homologous chromosomes, okay? Anytime a question has the word homologous chromosomes in it, you have to be in meiosis 1, because that's the only time that you have homologous chromosomes. So if you get a question that says the homologous chromosomes line up on the middle, well, I'm hearing middle, so I know it's either metaphase 1 or metaphase 2, but because it says homologous chromosomes, that tells me it's in metaphase 1. What if I say, okay, now the homologous chromosomes are being pulled apart from each other? Well, we know that apart is anaphase, but how do you know which anaphase it is? Well, because I said sister chromatids, homologous chromosomes, because I said homologous chromosomes are getting pulled apart, that's how I know it's in anaphase 1. So in meiosis 1, we're dealing with homologous chromosomes. In meiosis 2, we're dealing with something called sister chromatids. So now look at this picture over here, prophase 2. Notice how these cells only have 1 chromosome, right? That's 1 chromosome there, and up here it's 2. So the key word for meiosis 2 is sister chromatids. So if I say something like the sister chromatids get pulled apart, well, I know that that's anaphase 2 because I said sister chromatid. In meiosis 1, it's homologous chromosomes that get pulled apart. So homologous chromosomes apart, sister chromatids apart down here. So if you can remember that, that'll really help you remember which one's which. Up here, all of these are homologous chromosomes. Down here, all of these are sister chromatids that are being lined up or pulled apart. Okay, so let's move on. Okay, so the next thing I wanted to talk about is this can arise, and this will cause some chromosomal abnormalities. It's known as nondisjunction. Basically what's going on here is that this is when the chromosomes don't separate properly. Remember earlier in an earlier lesson I told you how those spindle fibers control the chromosomes kind of like puppets on a string? Well, sometimes those strings don't work the way they're supposed to, and the chromosomes don't separate the way that they're supposed to. So let's look at what's happening over here on the left. So we've got this chromosome, we've got this cell it's trying to divide. This nondisjunction happens, and you can see, compare the second cell division down here, that middle row, you can see that the cells are not the same anymore. On the left, there's 6 chromosomes, right? 4 pink and 2 blue, and on the right, there's only 2, right? And when we end up at the end of this whole process, now we've got cells with the wrong number of chromosomes. And so this could arise, this could be the reason why somebody either has too few or too many chromosomes. If you get one of these, you're going to have too many. If you get one of these, you're going to have too few, because the nondisjunction made it where they didn't separate properly. This is nondisjunction in the first cell division, but you can also have nondisjunction in the second cell division, and it works out where it's basically the same, but maybe not quite as bad over here. So now, in the first cell division, everything's separated properly, right? You can see that each cell got 1 pink and 1 blue chromosome. So pink and blue, okay? But now in the second round of cell division, the nondisjunction happened. And so now these 2 cells are not correct. This one has an extra chromosome, this one is missing a chromosome, but the 2 on the right are actually normal. Those separated properly. And so this just kind of shows how getting a chromosomal abnormality sometimes is just luck of the draw, right? You can't really blame somebody for it because they didn't have anything to do with it. If somebody is missing a chromosome or has too many, it's just a process of these chromosomes not separating properly, and obviously shouldn't be held against them because something happened to them that they couldn't control, right? So nondisjunction, this is when the chromosomes don't separate properly, and it'll cause some chromosomal abnormalities, which we'll look at here in just a second. Another really important thing that happens this happens in meiosis but does not happen in mitosis, is something called crossing over. What happens in crossing over is when these homologous chromosomes come together, they'll form something called a tetrad, and what will happen is these chromosomes just kind of exchange some information. They trade genes. And so if you look at the picture down at the bottom, on the far left, we're starting with a fully blue and a fully purple chromosome. But when they form that tetrad, some of their genes get swapped, and now at the end, the blue chromosome has a little bit of purple on it, and the purple chromosome has a little bit of blue on it. This is a good thing. It increases our genetic variety, but this is also part of the reason why you and your sisters don't look, or you and your siblings don't look just alike. We're doing this on purpose. It's kind of like shuffling a deck of cards and just kind of making things more random. So now you get some of your dad's genes that maybe your siblings didn't get, and vice versa with your mom. So this is a good thing. It increases the variation in our population, and it's why you and your siblings don't all look just alike. Okay? Great. So the next thing I wanted to do is take a look at a couple of these pictures. This picture is called a karyotype. What's going on here is that these are all the chromosomes in a cell for a person. This is a human, because you can see that there are 23 chromosomes here. In a karyotype, we always take the chromosomes, we align it with its pair, or the similar length chromosome, and then we organize them from the biggest chromosomes to the smallest chromosomes, except for the sex chromosomes always come last. Okay, and there's a couple things I need you to remember about the sex chromosomes. So they're always the 23rd pair, so they always come last. And then depending on what type of sex chromosomes you get, that is going to determine the gender of that individual, right? So a little mnemonic device I like to tell people is that if you have a Y, you are a guy. That's a really easy way to remember. If you have a Y chromosome, biologically you're a male. And if you have two X chromosomes, biologically you are a female. Now, it's not always that simple, as we'll take a look here in just a second. Okay, so again, this is called a karyotype. Let's take a look at this karyotype and see if we notice anything kind of odd about it. Okay, so it looks like there are not exactly 23 pairs, right? There's 23 pairs plus 1. So instead of 46 chromosomes, this is 47 chromosomes, right? Again, a karyotype, it's all the chromosomes listed out from largest to smallest, except for the last chromosomes are the sex chromosomes. And this individual has 2 X chromosomes and a Y chromosome. So my little mnemonic device does hold up here. If you have a Y, you are a guy. So this would be a male, but he has an extra X chromosome. This disorder is known as Klinefelter syndrome. And what happens is this would be a male who their body doesn't make as much testosterone as it normally would if you only had 1 X instead of the 2. So this individual, their body is going to make a little less testosterone, and that means that some of the characteristics are going to be less masculine. They do tend to be pretty tall, but they won't have facial hair, and a couple of other key characteristics there. But this would be a male, even though they do have 2 X chromosomes. So it can be a little tricky, right? Okay, so here's the last thing that we'll look at. This is another karyotype. So we have all of our chromosomes here. They're listed from smallest to biggest, and we can see that those sex chromosomes are at the end. Again, if you're XY, you would be a guy, and XX would be the female. What's going on here is this is a disorder known as trisomy 21. If you look right here at the 21st chromosome, they have an extra 21st chromosome. Instead of just 2, this person has 3 21 chromosomes. So we would call this trisomy 21, but what you probably know it as is Down syndrome. So this combination of this combination of chromosomes, this chromosomal abnormality would result in Down syndrome, and again, this is a result of that nondisjunction. Okay, so y'all, thank you so much for making it here today. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.